Measure twice, cut once, the old adage goes. An inch here or there can make a huge difference in a construction project. But when it comes to building a human body, those measurements go from inches to microns, 1/1,000,000th of a meter. Scientists at the UC San Diego stem cell program are working at a scale that small to answer some of life's biggest questions.
Our mission to understand brain development requires us to study the connection between the brain and the eye. Bioengineer Shaochen Chen is helping us thanks to his work connecting damaged spinal cords.
The human body is a massive network. Signals traveling along neural pathways, allowing us to see, smell, hear, and move. But what happens when those connections are severed? Can they be repaired? Or are they lost forever?
For the case of spinal cord, imagine we can using this kind of 3D printed live tissue for that patient, and they can get recovered within a couple of months and walk again.
Shaochen Chen is the founding director of the Biomaterials and Tissue Engineering Center at UC San Diego. His lab creates functional biological tissue using stem cells and a complex 3D printing process.
In bioprinting, we print cells with gel, and then we build a functional biologically active materials. So in the case of heart, when you print this tissue, it can beat. It's not just a plastic model. We have been working on this topic for the last almost 20 years. We start from scratch, and we build our own printing machines.
Those machines shine light into a gel full of stem cells using patterns derived from CT scans to match liver, heart, and nerve tissue. Any spot the light hits becomes solid.
These cells are from patients' own skin, so you can take these cells and reprogram them into functional cells like heart cell, liver cell, nerve cells. And then we can print these cells into the structure, and in that case, the piece is the patient-specific. And you can put it back into the patient. Hopefully it will help to grow the heart, repair the livers, and also regrow the nerve tissue.
Chen's lab has already found success repairing severe spinal cord injuries in rats. Now Chen's partnering with Alison Watry and Carl Wallen to help them understand how brain and eye development influence each other. He's devised a way to keep the tiny brains and retinas they're studying separate, just like they are in the womb.
It's a very exciting opportunity for us to work together with Alison's lab and Carl's lab. All three of us are maximizing our strengths to build this complex organoid system that has the retina in one end and a brain on the other end. Then there's a channel with the biomaterial that we studied over the years to bridge these two together in a very precise fashion.
That channel allows the brain and retinal organoids to form connections, so researchers can see how those connections influence brain development. And the Watry Lab is also hoping to use Chen's bioprinting techniques to create tiny blood vessels, allowing brain organoids to become larger and more complex.
In our case, we use a light, and it can really focus the light into a tiny spot or patterns, and then we can recreate this very fine vasculature network.
And bioprinting has other revolutionary possibilities. From speeding up drug testing,
it takes average 12 years and $2 billion for one drug. Now, since we can print this human tissue, they can directly test whether this drug will be toxic or whether this drug will be effective or not in the situation of the human tissue.
To perhaps one day patching the human heart.
Imagine yourself in the future that we can create this functional human tissue that can be used as a patch to put on the patient and repair the heart damage, and the heart go back to normal. And I think that's very exciting for me, and I'm so excited, to be involved in this kind of research work.